Hello everyone. This is a video in continuation to our videos for 10th CBSE where we had started the chapter number 6, life processes. In our previous video we had discussed about photosynthesis, how it happens and all. In this video we'll talk about a very important topic which comes in the exams, very important. One or the other experiment is there in the paper. So these are the various experiments on photosynthesis. There are 3 experiments which are given in your book, but we will be doing 4 because one they have asked you to do. So we'll be doing those 4 experiments one after the other. So please watch carefully and learn. First experiment. First of all, I'll just write what experiments are there, then we'll talk about some common concepts and then we'll start with the experiments. So the first would be to prove. Proof is for all the experiments. So the first one is to prove that chlorophyll is necessary for photosynthesis. Second experiment is to prove that carbon dioxide is necessary for photosynthesis. And third experiment is to prove that sunlight is necessary for photosynthesis. And the last one would be to prove that oxygen is produced during photosynthesis. Fine. So these are certain experiments which we have in our book for the process of photosynthesis. Let's do them one by one. Before we start with any of these experiments, there are 2 steps in all these experiments which are common. Whether we are doing first, last experiment, any experiment, these steps remain common for any of these. So the first step is destarching. And the last step, that is iodine test. First is destarching, last is iodine test. Whether we are doing any of the experiments, we have to destarch the plant first, as in the plant is kept in dark for 24 to 48 hours to destarch it. Destarching ka matlab, whatever starch is present inside the plant, that is used up by the plant and now the plant has no starch. So that we can understand, like for example, एक plant है, उसको हम पहले destarch करते हैं and then we perform the experiment और हमारे पास एक result आता है. तो हमें पता है कि whatever starch is formed, that is formed during the experiment. Previously, there was no starch present in the plant. So to make sure that whatever starch is formed in the plant, that is formed during the experiment and there was nothing present before the experiment. For that we destarch the plant first. How is it done? It is done by keeping the plant in dark, somewhere dark where no photosynthesis can happen for 24 to 48 hours. This is how we destarch a plant. Last step is iodine test, which will be performed for any of these. So for iodine test, एक normal जो हमें पता है, that is iodine is put over starch, it turns blue black. So what are the steps which we follow here? We take the leaf on which the starch test or iodine test has to be performed. We keep that leaf or boil that leaf in water first. Please listen carefully. We boil it in water to kill the cells of the leaf so that the cell wall disintegrates and the cells of the leaf are denatured. Then we'll take it out and put it in alcohol and boil it in alcohol. When we boil it in alcohol, chlorophyll is removed. So the chlorophyll is destarched when we put it in alcohol. After that, the leaf is spread and iodine is put over it. The parts which contain starch turn blue black and the parts which do not have starch do not change color. So this is our iodine test. Steps of iodine test are very important. You must know all the steps. In your book, they have just said, why do we boil it in water? Why do we boil it in alcohol? So you write in front of it that we boil it in water to kill the cells. We boil it in alcohol to remove chlorophyll and then iodine is put. When iodine is put, the parts which have starch turn blue black and the parts which do not have starch remain colorless. So these are the 2 steps which will be same for any of these experiments. Let's start with the first one. That is chlorophyll is necessary for photosynthesis. That is experiment number 1. Now, whenever we have to prove that whenever this question comes in your paper, they are going to give you 2 leaves wherein variegated leaves are there. So this is the speciality of this experiment, when we have to prove that chlorophyll is necessary for photosynthesis. What do we need to do? We need to select a plant which has green and non-green patches in it. As in, suppose this is a yellow patch and the rest of the leaf is green. This is also yellow, this is also yellow. So a leaf, such type of leaf which has green and non-green parts in it, is known as a variegated leaf. For example, in money plant, croton, we have these type of leaves. So we have to select a potted plant, a plant which is there in pot and has variegated leaves. Now what do? We take that plant, keep it in dark for 24 to 48 hours, destarch it. Now we take it out and keep it in sunlight for a while. When we keep it in sunlight for a while, after that we pluck one leaf and perform iodine test. How do we perform iodine test? We'll boil it in water first, then boil it in alcohol and then put iodine over it. What do we observe? That the parts of the leaf which were yellow in color, they do not change color, and the green parts, they turn blue black. Why? Because the green part was containing chlorophyll and it had performed photosynthesis, and hence the color changed to blue black, whereas the yellow part had no chlorophyll, so no change in color took place in the yellow portion of the leaf. So this is an experiment to prove that chlorophyll is necessary for photosynthesis. Let's see the second experiment. Second experiment is to prove that carbon dioxide is necessary for photosynthesis. What do we do here? For this experiment, we take a normal plant. We don't have to take a variegated or any other type. We take a normal plant, and what do? Again, we take that pot, keep it in dark for 24 to 48 hours and take it. Matlab we have destarched. After destarching, what do? We keep this plant here, and over this plant we will place a bell jar, and inside this bell jar we will place a petri dish which will have KOH crystals. What is the property of KOH? Whenever you get a diagram in the paper and questions from it and KOH is placed, you must understand that this is an experiment to prove that carbon dioxide is necessary for photosynthesis, because KOH has a property to absorb carbon dioxide. It absorbs carbon dioxide. So what will it do? It will absorb the carbon dioxide from this bell jar. So this leaf is getting sunlight, is getting water, has chlorophyll, but there is no carbon dioxide. So what do? We place it in sunlight for a while and then pluck one leaf and perform iodine test over it, boil it in water, boil it in alcohol and then put iodine. There is no change in color in the leaf because this leaf was not having any carbon dioxide, so did not photosynthesize. So it proves that carbon dioxide is necessary for the process of photosynthesis. Fine. So this is the second experiment. The third experiment is not there in your book. They have asked you to frame this experiment to prove that sunlight is necessary for photosynthesis. What do here? We take a normal plant, as in no variegated leaves, a normal plant. We place it in dark for 24 to 48 hours. Take that plant now, and on one leaf of that plant, we will place a paper strip with the help of paper clips. उसके आगे और पीछे की तरफ एक पेपर स्ट्रिप लगा देंगे and then we will place it in sunlight. After some time we'll pluck this leaf where the paper was placed and then perform iodine test over it, boil it in water, boil it in alcohol and put iodine over it. What do we observe? That the part which was covered with paper, it shows no change, and the part which was exposed turns blue black, which shows that the part which did not get sunlight did not show any change in color, whereas the other part of the plant, it has turned blue black. So this proves that sunlight is very important for the process of photosynthesis. So these are the three experiments from photosynthesis from your book. In our next video, we will be talking about the next topic, that is heterotrophic nutrition. We will talk about the type of heterotrophic nutritions, and then we will discuss nutrition in amoeba and paramecium. And uske baad in the upcoming videos, we will talk about the digestive system or nutrition in human beings. So keep watching, liking, sharing, subscribing. Thank you.